Hello, everyone, and welcome to today's live broadcast, Cas9-Based Genome Engineering. It is presented by Alejandro Chavez, MD, Ph.D., and Assistant Professor of Pathology and Cell Biology at the Columbia University Irving Medical Center. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Chavez. I will now turn the presentation over to him.
Hello, my name is Alex. Happy to be here today, and I'm currently Assistant Professor at Columbia University. I'd like to share with you my work on Cas9-Based Genome Engineering. So for the overview of my talk, I'll tell you first what is Cas9 to make sure that we're all on the same page. I'll then tell you some of our lab's innovations with Cas9, including our work on building Cas9 activators, tuned guide RNAs, and then our guide plus donor strategy. So what is Cas9? Cas9 is a nuclease that's directed to DNA via a small little RNA. The RNA is called a guide RNA, and the guide RNA contains 20 nucleotides of complementarity towards the target site you want Cas9 to go towards. Cas9 and the guide RNA then complex together and they'll bind DNA. Once Cas9 and the guide RNA are bound to DNA, Cas9 will cleave both of the DNA strands. So what else can you do with Cas9 other than cutting DNA? Well, if you mutate the residues within the protein that are involved in that DNA cutting, you can make what's called dCas9 or dead Cas9. So dead Cas9 is still able to bind DNA, but it won't cut it. And so in the lab, we kind of equate dCas9 to a molecular couch potato. So go wherever you want, and it'll sit there like a lump, and you're just not going to do anything. You can then direct this little couch potato upstream of a given gene's promoter, and by fusing various effector domains to dCas9, such as domains required for gene activation or for gene repression, you can actually now regulate that gene's transcription. So why should you care about dCas9, and in particular dCas9 activators? Well, they're really useful for understanding fundamental biology. So let's say you want to look for suppressors or enhancers of phenotype. In this case, imagine you have a fly, has a normal eye morphology. To express a polyglutamine tract protein within the eye, you can see the eye loses a lot of its morphology. And then if you want to look for suppressors of that phenotype, you can use Cas9 activators to try to find one. In this case, if you overexpress HSP70 within those eyes that also express the polyglutamine protein, you see a nice rescue of phenotype. In addition, activators let you model disease. They let you model things like gain-of-function mutations within genes or gene amplification, such as occur in cancer. And then finally, they're very useful for things like cellular differentiation. So in this case, imagine you have an iPS cell and you want to convert it to different sorts of neurons. That a lot of times requires multiple transcription factors, and you can imagine using Cas9 activators to turn on whatever selective portion of transcription factors you want to allow for that differentiation to occur. So when we started this project, what was the current state of Cas9 activators? Well, they were able to activate, but they weren't very potent. And so here I'm showing you 10 different guide RNAs, each against the same gene. And what you'll see is that most of them turn on the gene at most like twofold, threefold. And so you can actually get more activation if you combine several of them together, but even doing that, it wasn't very good. And at most, you still got between 5 to 50-fold gene activation. So what an ideal Cas9 activator, what would that look like? Well, you'd want it to work with just one guide RNA. And that's important because it lets you do things like genome-wide screens. In this case, give one guide to any given cell you want, and then look at the phenotype. And also, you want to do robust gene activation. Turn on gene 1.1-fold is probably not super useful to you. And so we chose an ambitious goal of about 100-fold induction for any given gene that we chose as kind of like our goal for the project. So what was our experimental setup? We identified transcriptional activation domains from the literature. We then fused them to the C-terminus of dCas9, and then we tested these fusions in a fluorescent reporter assay. The way the assay works is you have a TD tomato fluorescent reporter gene, which is downstream of a minimal CMV promoter, and then it has a guide RNA binding site upstream of that. We then direct dCas9 plus the activator to that reporter, and if it's able to turn the reporter on, our cells turn red, and then we can quantify the amount of redness using just flow cytometry. And so what did our results of our screen look like? On the y-axis, I'm going to quantify for you mean fluorescence intensity. On the x-axis, I'm showing you all the different fusions we tried. And what you see is that here's the data, and that really only 3 fusions seem to work very well. And so the conclusions from this experiment were that most fusions failed, not that exciting of a conclusion, that VP64 was a really, really good activator. And that led to some problems because the issue was that we needed to improve upon VP64 because at the time we started this project, VP64 was the known best activator. I was just surprised to find that it was so good because something that was invented in the 80s or 90s, I thought there's no way it could be that good. That was wrong. So basically, after we got these results, we decided to look at how genes are activated naturally to see if we can get inspiration from that to maybe figure out how we can improve our system. So how does endogenous transcription work? So in native systems, you actually have not just one transcription factor binding to a promoter, you actually have dozens binding to that given gene's promoter. And then those transcription factors each interact with a different portion of the transcriptional machinery. And by doing so, they each cooperatively recruit RNA polymerase. So for example, if you have a promoter that binds 2 elements, you'll get very weak transcription. If you have a promoter that binds 4 different transcription factors, they're going to pull better on RNA polymerase, and thus you'll get more transcription. So we had a very simple hypothesis, and that was, can we mimic this cooperative recruitment by simply fusing more activation domains to Cas9? So what does that look like? So we started with our 3 best activation domains, also kind of known as the only 3 that actually worked, which are VP64, P65, and RTA. And then we used the same fluorescent reporter assay. This time on the x-axis, I'm going to quantify for you median fluorescence. And on the y-axis, I'm showing you some of the initial single fusions we tried. And so you see that they do activate the reporter a little bit. But when we fuse them in combination or as a full triple, we saw a much improved activation. There's about a 10-fold difference between VP64 alone versus VP64 and the 2 other elements fused to it. And so this was really promising, but this is just a synthetic target reporter gene. So we wanted to see what this looked like for endogenous genes. So for our endogenous target, we decided to use VEGF. And so we once again repeated the same experiment. This time I'm going to quantify for you VEGF induction on the x-axis. And once again, the single fusions, they were able to turn a little bit of VEGF on. The double fusions were even better. The triple fusion was rather good. And so once again, it was about a 10-fold difference. And so for the remainder of the talk, I'll refer to this VP64, P65 RTA fusion as VPR or Viper, just because it's kind of cool. So we tested other genomic loci. We tested a few others like NeuroD1 and ROXF2. These are both transcription factors. And we compared our activation with VP64 versus Viper. And what you see in both cases is that Viper is significantly better than VP64. It's about 20 times better. And to date, I've tested, I don't know how many dozens of loci, and Viper basically is always better than VP64. So activating one gene at a time is really nice. But if you come from the church lab, which is where I did my postdoc, George always likes to ask you, like, have you multiplexed? And so the reason why multiplex is important is because cells don't actually just do one thing at a time. When they see a stimulus, they respond with many different things. So, if they see heat, they don't just turn on one gene, they turn on whole families of genes. And so if you ever want to be able to actually ask that level of biology, we need to have tools that let us do it. And so to test multiplexing, we targeted 4 genes at once. And these are the 4 genes on the x-axis. And when you activate them with VP64, you see they get turned on a little bit. You activate them with Viper, they get turned on a lot more. It's about a 10 to 30-fold difference between them. And so summary for part 1 of my talk, we designed a new synthetic activation domain. And this led to about a 10 to 30-fold improvement over the previous best activator, VP64. In data that I didn't show you, we showed that our tool works not only in human cells, which is where we did the majority of our experiments, but it also works in mouse cells, fly cells, and in yeast cells. And finally, we actually, at the end of our story, we used our Cas9 activator to convert stem cells into neurons, just showing you can actually get nice functional phenotypes using our tool. And this paper was published a while back in Nature Methods. So the second part of my talk, I'll tell you our work on tuned guide RNAs. So tuned guide RNAs endow Cas9 with single nucleotide specificity. And we actually can use our tuned guide RNA strategy to do something which is a little bit unique, which is prevent mutations from emerging within populations of cells. So let's talk a little bit about Cas9 specificity. So Cas9 is a really powerful tool, but the problem is that it has difficulty discriminating between 2 highly similar targets. So imagine you have 2 alleles. One's a bad allele, and another's a good allele. They differ by a single nucleotide polymorphism. And what you want to do is you want to have Cas9 cut the bad allele, but you want it to leave the good allele alone. And so you say, no problem, I'll just give the cells a guide RNA that's perfectly complementary to the bad allele, and what will happen? Cas9 will cut it. The issue you face is that that same guide RNA that's perfectly complementary to the bad allele will still have only one mismatch to the good allele, and likely Cas9 will still cut the good allele. So how do you improve Cas9 specificity? So we started thinking about this problem. We realized that Cas9 certainly tolerates single mismatches fairly well. We then wondered if there are more mismatches, if this would then actually inhibit Cas9's ability to cut. And this was kind of known in the field. And so we said, could we exploit this difference where more mismatches eventually peter out Cas9 activity to actually somehow tune Cas9 to have single nucleotide specificity? So we said, what if we started with a guide RNA that wasn't actually perfectly complementary to the bad allele? So let's say it had one mismatch to the bad allele. Well, once again, so here's a single mismatch. Once again, Cas9 probably won't mind that mismatch, and so likely it'll cut the bad allele. On the flip side, that same guide RNA actually has two mismatches against the good allele. It's got one that's the common mismatch, that's mismatched on both targets by, and the second one, which is the allele mismatch, that's unique to the good allele. And so we wondered if Cas9 would then actually have trouble interacting with the good allele and actually not cut it. So the question now was, does our deal really work? And so to test that, we made two variants of the TEM1 beta lactamase. And so one variant was S68, and the other was N68. And these two alleles differ by single nucleotide polymorphism. We then created a library of guide RNA variants such that each of them had a single mismatch to the S68 allele, and thus by definition would have two mismatches to the N68 allele. Okay. And so we then screened our library, and we're hoping to find guides that would cut the S68 allele, but leave the N68 allele alone. That was our goal. And so when we did that screen, we identified several guide RNAs that seemed to have that property from our screen. And we decided that seemed to be active on S68, but leave N68 alone. And so we decided to validate our results in E. coli. And so what we did was we gave cells either S68 or N68 alleles. We then fed them either a control guide, which targets nothing inside of the E. coli genome, or what we call an untuned guide. This is a guide that is perfectly complementary to S68, and by virtue of that only has a single mismatch to N68. Or our screen guides. These are guides that have one mismatch to S68 and then two mismatches to N68. And after we threw those into cells, we quantified cell growth. And what's important for this assay to know is that if the guide RNA cuts the alleles, the cells grow very poorly. And so what you see is that the control cells, they grow very well because the guide doesn't touch either of the alleles, so it leaves them alone. They hit nice density. If you feed cells an untuned guide, you see that both S68 and N68 alleles both die. And that's because the guide basically kills them both, doesn't really discriminate between either of them. But if you feed cells one of the screened guides, you actually see that the S68 allele sees a marked growth deficiency, while the N68 allele containing cells don't seem to mind it. And this hints that our system is actually specific for cutting S and leaving N alone. And so we named these screen guides tuned guides because they're for their high specificity, because they're tuned to a particular target. So how can we use this system for mutation prevention? So we started thinking to ourselves, if tuned guide RNAs really did give Cas9 single base pair resolution, you could actually imagine designing a system such that it's benign to wild-type cells with normal genomes, but when a cell gets an undesired mutation, it's designed such that Cas9 will cut those mutant alleles away from the population. And by doing so, prevent those mutations from ever becoming fixed in the population. So do tuned guide RNAs actually enable mutation prevention? To test that, we loaded cells with our TEM1 N68 allele. And then what we did is we grew them up overnight. And what's important to know is that some of the cells randomly after overnight growth, they'll convert the N68 allele into the S68 allele. And when they do that, when you get the S form of this allele, it actually causes the cells to become resistant to ampicillin. And so we can simply take cells, grow them up overnight, plate them to ampicillin plates, and just count the number of AMP colonies. And that's a mark for how many times the N allele converted into the S. And so what we did was we loaded cells with either our control guide, and we got a lot of revertance overnight, or we loaded them with a guide that was tuned and selective to only destroy S68 alleles but leave N68 alleles alone. And after overnight growth, what we found is basically we got 1,000-fold fewer colonies, arguing that our system was able to actively cut any S68 allele that emerged from the population. And so this was really promising, but again, it was only a single target, so we needed to do more. And so to cut to the chase and cut out a lot of experiments, I'll just show you kind of a really nice big picture experiment where we did multiplex mutation prevention. So in this case, we took our tuned guide RNA strategy, and we designed it to simultaneously prevent a total of 4 different mutations in 2 different genes all at once. And so for on the y-axis, I want to quantify for you mutation frequency. And then I'm showing you what the mutation frequency of these different alleles coming out randomly in a population are with cells that just have control guides in them. So there's no system trying to prevent their occurrence. And what you see is that some of these alleles are very frequent, occurring in almost 60% of the cells. And when you then load a cell with the tuned guide RNA system that's designed to prevent each of these 4 mutations all at once, you basically see that you don't get any of the mutations emerging from the population. It basically prevents them all, essentially to the error-rated DNA sequencing that we use in these experiments. So summary for part 2 of my talk. Tuned guide RNAs allow Cas9 to discriminate between highly similar targets. They let you cut that bad allele while leaving the good allele alone. And you can actually use these tuned guide RNAs to enable a new form of evolutionary control, which we like to call mutation prevention. And so basically lets you kind of prevent any mutation from becoming fixed in the population once you've designed a system against it. And this paper was recently published also. All right. And for the final part of my talk, I'll tell you about our work on our guide plus donor strategy. And this is really a method for allowing us to simultaneously make hundreds of cells with programmed mutations all at once. And so a little bit of background, as you all know, the world is just becoming flush with sequencing data. Worldwide, sequencing capacity seems to be doubling about every 7 months. And right now, we've sequenced less than 1% of the human population. But in doing that, we've already identified over 300 million human single nucleotide polymorphisms. Of those, about 20 million are predicted to cause amino acid changes within proteins. And so those are obviously going to be influencing our phenotype in ways that we still don't actually know, nor are we able to really predict very well. In addition, that's just in humans, but as we've been sequencing more of other species, we're also finding hundreds of millions of SNPs within them. And so the issue is, what the majority of these SNPs do, no one has a clue, and we really need to start building technologies to help us address those sort of questions. So what is the gold standard for testing the significance of a SNP? Well, one is you take a wild-type cell, you then introduce the mutation into that locus to make the mutant you're trying to study, and then you compare the mutant cells and the wild-type cells, and you hope that the mutant cells have some phenotype that's different from the wild-type cells that you can now attribute to that mutation. And so how is it that people make these cell lines nowadays? Well, of course, they use Cas9. And so how do you use Cas9 to make these cell lines? It's pretty straightforward. You take cells, you give them Cas9, you direct Cas9 to your locus of interest. Cas9 then binds that locus of interest, and it cuts the locus. Then if you have fed the cells at the same time a donor DNA molecule that can be used to repair that break, the cell will grab that donor and use it as a repair template. And by doing so, it'll incorporate your change into the site. So what are the issues with Cas9 engineering? One, while Cas9 is certainly powerful and is probably the best tool we have out there right now to make genome modifications, current approaches are very slow because they basically do one mutant variant at a time. So you take your time, you build one cell line. After a couple of months, you then test it. And so that's really labor intensive. It's also rather costly, even though it's much obviously cheaper than it was several years ago. And in addition, once this clone is generated, you then are going to test them also one at a time. And so at this rate, if we're building mutants one at a time and we're testing them one at a time, we're all going to be long gone before we can test the hundreds of millions of SNPs that exist out there, and the hundreds of millions, maybe billions more, we're eventually going to discover over time. So how do we bridge this huge gap between the amount of mutations we're finding on sequencing data and the amount we actually understand and we want to test in the lab? So to begin to systematically catalog the effects of genomic variants, we need scalable methods of generating thousands of mutant cell lines at once, and also tracking their fitness within a pooled fashion. Because it doesn't help us make millions of cell lines at once, but if I have to test them one at a time, that's not super useful either. And so as a first step towards this goal, we decided to focus our technology on a very simple model eukaryote, and that's yeast. And we decided to see, can we use yeast to build this sort of technology that would let us make thousands of mutations all at once and also track their phenotypes all at once. So the first thing we need to address is why is it that current methods actually don't scale to these hundreds of thousands of mutations at a time? And so when you're trying to make one single mutation in a cell, you are going to take a guide RNA against the locus you want and then feed it the donor against that locus that you want to have it used as a repair template. And the cells will likely that get one guide RNA will probably get also a little bit of the donor. And that lets you have a very high frequency of targeted mutation. So that's good. It makes it very efficient. But now imagine you have a pool of guide RNAs and a pool of donors. So each guide has a unique donor associated with it. Well, now just because a cell gets one guide, it may not actually get the proper donor with it. Because of that, your frequency of getting the proper mutation you want is actually very, very low. So how is it that you prevent the uncoupling between the guide RNA and the donor during transfection? Well, the answer is rather simple. Why not try to fuse them together? And so what if you fed cells a library of plasmids such that each library contained a unique guide RNA and a unique donor that were fused in cis? So basically, every single guide RNA had its appropriate donor template right with it. Then you could feed these guide plus donor plasmids to a Cas9 expressing cell line. The Cas9 cell line would get each one of the guide plus donor plasmids. And now, because the guide and the donor are coupled, the mutation frequency would be very high, and you get a very efficient conversion to the mutations you wanted. So we call this strategy our guide plus donor strategy, and we'll describe how we tested it next. So in addition, what are other uses for the guide plus donor? Well, because every single mutant cell line carries a unique guide plus donor plasmid, by sequencing the prevalence of a given guide plus donor within the cell population, you can actually track its fitness. So imagine you have a starting population of cells where 2 of them have red guide plus donors, 2 have green guide plus donors, 2 have yellow, and 2 have blue. You then subject those cells, that mixture, to your experimental condition, and you get out the library at the end. In this case, you see you have 2 reds, 5 yellows, and 1 blue. Well, what this means is that by sequencing your starting library and your ending library, you can actually then understand what each of the mutations did for fitness. And so, for example, at the start of this experiment, we had 2 green cells. At the end of the experiment, we had no green cells. And so what that tells you is that the green mutation introduced is deleterious under your experimental conditions. And so simply by sequencing the guide plus donor, you kind of can figure out the fitness of all of your cells, which is really useful. So how well does our method work? Well, we use our guide plus donor strategy to introduce point mutations into a series of loci. And we tested 3 different loci, one at a time. RS214, which is a non-coding region, or SGS1 and SRS2, which are both coding regions in the yeast genome. And I'm going to show you on the x-axis the efficiency of conversion. And what you see is basically we have about 80% efficiency of conversion when we use our guide plus donor strategy, with 80% of the colonies arising post-transformation having the mutation we expect them to have that we designed. We also saw similar high efficiency when we were trying to perform short 60 base pair deletions within the genome or short 15 base pair insertions within the genome. So this kind of told us that our strategy seems to be pretty plastic, and you can do more than just point mutations. You can do things like small indels, which are also very frequent in the human genome that many times you actually want to understand also. So introducing one mutation at a time works, which is great. Now the question is, can we make several mutations all at once in a mixed pool? And can we track each of the mutant's fitnesses simply by sequencing out the guide plus donor fragment? So to test this, we started with a small library of guide plus donor plasmids. They were directed against either the RS214 non-coding, which is kind of our control region of the genome, or SGS1, which is a protein involved in DNA repair. We then pooled the guide plus donor plasmids against both RS214 and SGS1 together, and we did a pooled transformation. We then grew the colonies up either overnight in regular conditions or overnight in the presence of a DNA damaging agent, such that cells that are mutant for SGS1 should grow more poorly in the presence of a DNA damaging agent, while cells that were targeting against the non-coding region shouldn't really care. We then just sequenced the prevalence of the guide plus donor plasmids within each pool, the ones that grew up overnight in regular conditions versus the ones that grew up overnight in DNA damaging conditions. And then we compare the guide plus donor abundances between different conditions. And so what did our data look like? So in gray, I'll show you the RS214 mutants. In red, I'll show you the SGS1 containing mutants. And on the y-axis here, I'm going to show you the log 10-fold change in cells that grew under drug conditions versus no drug conditions. And so what you see is that cells that had mutations against SGS1 that were supposed to abolish its activity, they saw a marked decrease in their fitness when grown in drug conditions versus non-drug conditions, versus cells that had guide plus donors that are targeting RS214, which should have no effect on DNA damage responses. They tended to grow much better. And so once again, and also we looked at the cells that had deletions in SGS1 or replacements in SGS1 that were supposed to basically pull a piece of SGS1 out and insert kind of junk instead of it, also grew poorly, as you would expect, since they're all disrupting SGS1 function, while doing the same deletions or insertions into RS214 didn't have an effect. And so the thing you might be asking is, well, Cas9, you're directing Cas9 to cut SGS1. Maybe all that's happening is SGS1 just getting cut and destroyed, and that's why all these cells are growing more poorly in the presence of DNA damaging agent. It has nothing to do with the fact that we're actually making the changes we want to make. And so to test that, we did what we call a recode. And so basically we had a guide plus donor that was designed to cut SGS1, and rather than disrupting the allele, simply would change the coding for the amino acids. So it wouldn't alter the amino acid composition, it would just change the DNA sequence for them, introducing only synonymous mutations. And what you see is that the recoding of SGS1 had no effect on DNA damage sensitivity, arguing that our system is actually specific for not only cutting SGS1, but also replacing it as we intend. And so this was really great. We were excited that our mixed pool would work. And so now the next thing to do is see what kind of fun things we can do with this sort of technology. And so one of the first things we did was we did a tiling deletion series along SGS1. And so we made a library of 60 base pair deletions within SGS1 along the entire gene. And so on the bottom here, I'm showing you what the SGS1 gene looks like, and has several different domains that are important for its activity. One, it's got a top 3 binding domain that's required for it to interact with the essential topoisomerase type 3 protein, which is required for SGS1 to do its function. There's a domain within SGS1 that actually is the helicase domain that's required for SGS1 to be a helicase and serve its functions in DNA repair. And finally, there's an RQC domain, which SGS1 uses to bind different DNA structures during its roles in DNA repair. And as blue dots, I'll show you guide plus donors that were designed to delete 60 base pair fragments from the SGS1 genome at different portions of the gene. And then on the orange dots, I'll show you guides that were against SGS1, but instead of having a donor that was supposed to replace a piece of SGS1, they just had junk donors. So essentially, these should kind of like do nothing to the cell. And so on the y-axis, I'm going to show you log 10-fold change in cells that are growing in drug versus no drug condition. And once again, what we see is that there are the blue dots. There are some of them that get depleted. In particular, the regions of SGS1 that correspond to the top 3, the helicase and RQC binding domain, they seem to be less fit when deletions are caused within them versus deletions with any other part of the protein, which is kind of known from previous literature. In addition, the guides that were against SGS1 but didn't have a donor to cause a deletion, basically they didn't see any fitness effects between the drug and no drug condition. So what about making point mutations, which is another thing you want to do with our system? So to test that, we took SGS1 again, and we targeted several conserved residues within SGS1. And we changed all that conserved residue to all other residues, but for the sake of clarity, I'm just going to show you 2 of those changes. In this case, I'm going to show you data for converting the residues to either alanine or isoleucine. And then we tested the fitness of each of these mutants using a range of different DNA damaging agent concentrations, either the lowest amount, which is 5 millimolar of hydroxyurea, or the highest amount, which is 40 millimolar of hydroxyurea. And SGS1, by the way, is required for resistance to this drug. Once again, I'm going to plot for you log 10-fold change in drug versus no drug condition. And what we see, we basically get all the results we expect. And so to walk you through some of them, we see that if you add more drug, which is the dark blue dots, the cells tend to be more sick than if you add less drug. So this is expected because the cells that are mutant for SGS1 in these loci should grow less well as the amount of drug increases and you mutate these conserved residues within the protein. In addition, if you mutate the lysine 706, which is the critical catalytic residue within the helicase, as you see, the cells don't like it, and that's expected. You can change it to alanine, or which is usually kind of benign, or isoleucine. It doesn't really matter. The cells don't like it. You throw in DNA damaging agent, they're unhappy. If you change valine 958 to isoleucine, you see that cells don't really care, and that kind of makes sense because valine and isoleucine are highly similar to each other. If you mutate a charged residue to an uncharged residue, so you take glutamine 986 and convert it to isoleucine, you see that that's very deleterious. And so once again, basically, we kind of saw all the expected results you would anticipate when you did these mutations. And so it kind of helped us know that our system seemed to be working as we expected. So we had done a lot of experiments now on a single gene, but we were really interested in knowing, can you do this, use our sort of system on genome-wide scales? And so to test that, we generated a new guide plus donor library that was designed to basically mutate all 300 small open reading frames, or SMORFs, within the yeast genome. And so what are SMORFs? SMORFs are open reading frames that are less than 100 amino acids in length. What's funny is that classically, open reading frames had been defined as being 100 amino acids and larger, but that's an arbitrary number. There's no reason why a protein can't be shorter than that. And so actually, SMORFs tend to be rather neglected in the biological space. And so we thought this was a good opportunity to try to understand their function better. And so, like I said, they're poorly studied. But what's interesting about SMORFs is that over time, we're starting to learn that they actually have very important biological roles. So SMORFs actually some are known to actually be essential, especially in even higher level organisms. And so we thought this could be a really cool opportunity to actually start to understand what they might be doing. And so in addition to making a SMORF library, within that library, we also included several guide plus donors against both essential and non-essential ORFs. And so the first question we asked is simply which SMORFs, or are there any SMORFs, frankly, that are essential for viability in yeast? And so to test that, we transformed our library into yeast, and then we compared the abundance of the guide plus donor plasmids in cells that either did or didn't express Cas9. What we saw is that when you targeted non-essential ORFs, basically there is no depletion of any given guide plus donor and Cas9 expressing versus non-Cas9 expressing cells. And that makes sense because if you mutate these non-essential open reading frames, the cells don't really mind. And on the flip side, if you target essential ORFs, you see that there's a marked depletion in guide plus donors that are against those in the Cas9 expressing cells versus the non-Cas9 expressing cells, which is exactly what you'd expect. If you're targeting a gene that's essential for growth, they're not going to grow. And finally, when we targeted the SMORFs, we were kind of excited to see that there was a small subset of SMORFs that actually appeared to be essential for the cells to grow. While the vast majority of SMARFs, though, appear to also to not be essential. So still is an interesting result. So the next thing we asked is what other roles might exist for SMARFs. And so to do that, we subjected our library to multiple different conditions. We grew them at higher temperatures. We grew them in the presence of DNA damaging agent, or we treated them with the antifungal fluconazole. And what you can see is basically that SMARFs, there are mutants of SMARF mutants that basically had ultra-sensitivity in all these different situations, arguing that SMARFs actually play significant biological roles in a lot of different contexts. And so this really suggested that SMARFs really have global roles that are yet to be discovered, and that it's actually an interesting space of biology that we should really start exploring more. So summary for part 3 of my talk. Basically, there's hundreds of millions of unstudied SNPs out there. The guide flows donor strategy is a way to actually simultaneously generate hundreds to thousands of mutants at a time and actually track their fitness. And you can use them to either study a single protein, or you can use them to examine entire families of proteins across the whole genome. And we were lucky that this paper recently got accepted for publication in Nature Biotech and will be in press soon. With that, I'd like to thank a few people. So I've had some really amazing collaborators over my time. And in particular, for the Cas9 activator work, I'd like to thank John Scheinman and Sue Vora, which are a postdoc and a grad student in the lab, respectively, that really helped push that project forward. In addition, Ben Fruit was instrumental in the two guide-RNA work. He was a staff scientist at the VIS when I was there. And then Xiaoge Guo and Angela Tung were really critical in making the guide plus donor strategy as amazing as it became in the end. Xiao really did a good job of leading that project over the course of several years, because technology was not easy to get to work, as we would expect. And our current funding sources, I'd like to thank those also, which is Columbia University and also the Burroughs Wellcome. And with that, I'll take any questions.
Thank you, Alex, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. So let's get started. First question is, BPR appears to be one of several Cas9 activator systems. Is there a best system?
So I get that question basically every time I give this talk. And basically what happened is several years ago, I got tired of getting that question. And so what we did was actually we decided to compare all the different activators together. And we compared, there's about 7 to 8 activators now that have been published. There might even be more over time. I've kind of lost track of them, frankly. And basically when you compare Viper against multiple different activators, it tends to be one of the top 3 best. The 3 best are SAM, Viper, and Suntag. And actually, we ended up publishing this work because we thought it was really important. So there's a later paper that people should look at that's published in Nature Methods that has my name on it, so you can use that to find it. That basically kind of compares the different activators together. And we think that's currently the best data that exists out there to really help people answer this question of what's the best activator and which one should they use for their studies.
Thank you. Given your experiments, can you predict what mutations you need to introduce into a guide RNA in order to give it allele specificity?
Yeah, so this question is referring to the tuned guide RNA strategy. In that strategy, we introduce mutations into the guide, and by doing so, we hope to have it have specificity towards one allele but not another. And so obviously, one of the things that we would love to do is be able to predict what the mutation should be that we introduce into the guide to give it that specificity. It turns out from the amount of experiments that we've done, which is really we've only made about 5 tuned guides, I don't really think we're capable of predicting the rules. Other people look at my data and they think they know what the rule is, but I just don't feel comfortable making assumptions on an N of 5. It would be interesting to try to do a larger scale analysis, try to understand what the rules are, but at this stage we haven't.
Thank you. Have you tried your guide plus donor method with human cells?
Short answer is no. We literally just got the guide plus donor method working fairly well on yeast recently. One of the areas that we think would be interesting is exploring human cells, but I think there's a lot of reasons why it's going to be much more challenging for human cells. But as of now, we haven't tried it. I would anticipate it would work very poorly, but I don't know the true answer.
Okay, thank you. We have time for one more question. There are now multiple Cas9 mutants that have improved specificity. Have you tried to see if they give allele level specificity?
Yeah, so this is once again referring to the kind of mutation prevention stuff. So there's now, maybe there's 5 different enhanced Cas9 variants that I'm aware of. There's probably even more in literature. We had actually tried a couple of years ago 2 of the enhanced Cas9 variants to see if they would give us allele specificity similar to the tune guides. At least in our hands, they didn't. That's not to be said that they never could. There's now, like I said, additional ones, which I think would be interesting to test. And we kind of think that maybe like those enhanced Cas9 variants, I think, are very good for making Cas9 not touch things that have like 4 or 5 or 3 mismatches, but they may not be have allele level sensitivity. So we're kind of wondering if maybe in the future, there'll be some combination of tune guides plus enhanced Cas9 variants, which will let people actually hit that space easier and more reproducibly. But for now, at least what we've tried of the enhanced variants hasn't really helped solve the problem.
Thank you for that. I would like to once again thank Alex for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 31st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.